Biosensors. How synthetic biology can save lives. Consider this scenario. You're at a food market overseas, about to eat what looks like a tasty chicken stick, but your synthetic biologist friend whips out a small tube of bacteria and puts a piece of chicken into it. She then shakes the tube and it turns green, indicating that the food you were about to eat is contaminated with Listeria monocytogenes bacteria that can cause serious illness and death. There are often scenarios like this, where the early detection of specific substances could reduce the possibility of harm arising. Enter synthetic biology-derived biosensors. Biosensors are artificial biosystems that detect changes in the surroundings and produce a response relative to these changes. There are whole cell-based systems and cell-free based systems. Whole cell systems consist of a module that senses input signals and a reporter module that converts these into output signals. This takes place within a chassis cell, which could be bacterial, microalgal, yeast or fungal. Cell-free systems use aptamers, nucleic acids, peptides, enzymes, antibodies or proteins as probes to detect change and carry out this sensory operation within biochemical cell extracts. There is huge scope for biosensor applications in niches such as medicine and health, food and nutrition, and the environment. Biosensors could be used in medical and health diagnostics for detecting pathogens and metabolite imbalances, could be used for food freshness and nutrient level detection, and could be used in environmental sectors for the monitoring of contaminants and pollutants around mining sites, waste and sewage treatment plants and rainwater pipes, and for the detection of toxic substances before they accumulate to hazardous levels. For example, the detection of noxious gases and harmful chemicals before they react with biomolecules or accumulate in cells, resulting in DNA damage or severe oxidative stress. Biosensors consist of synthetic biological components known as bioparts, a sensing receptor or sensing biomolecules like DNA, aptamers, peptides, enzymes, antibodies and genetic circuits, a reporter or physicochemical transducer module, and chassis cells if the biosensor is a whole cell system. To make whole cell biosensors, a plasmid, phage, cosmid or artificial chromosome vector must be used to transfect a chassis cell. These vectors carry the sequences for any operators, inducible promoters, ribosome binding sites, genes and terminators. Sensing receptors are constructed from transcription factors, activators or repressors or sensor kinases of two-component systems in conjunction with regulatory elements. In activator-type transcription factors, the reporter genes are fused to the promoter of the transporter gene, which is regulated by the transcriptional activator. When a specific activating substance is present, the promoter is activated, and the reporter gene is expressed. In repressor-type transcription regulators, the reporter genes are transcriptionally fused to the repressor gene that's controlled by regulatory machinery. Sensors detect and or quantify a change and drives the reporter to create a response relative to the change. Colorimetric responses due to the production of a pigmented fluorescent or bioluminescent protein are usually ideal, as these are easy to identify. However, other responses include changes in metabolic activity, gene expression profile and pH. Advantages of whole cell biosensors are that the bacterial chassis cells replicate relatively easily and cultivation is inexpensive. Their use eliminates difficult and special analysis techniques needed to acquire results. Real-time monitoring can be conducted, and complex systems can be designed. Whereas the advantages of cell-free biosensors are that the purified proteins and antibodies used as sensors are very quick to detect substances. They do not meet the regulatory definition of a GMO. They do not replicate and are robust. Disadvantages of whole cell biosensors are that they are comprised of GMOs, they can have a short lifetime, and there may be difficulties in ensuring their robustness and reproducibility. The disadvantages of cell-free biosensors are that expression and purification conditions must be optimized, and their production and use is expensive. Current environmental biosensors can detect the following: heavy metal ions, for example arsenic, a wide range of structurally diverse antibiotics and toluene and flavonoid compounds aimed at reducing the rise in antibiotic resistance. neurotoxic organophosphates and nitrate bioavailability in aquatic ecosystems. Current disease-related biosensors can detect the tropical parasite Schistosoma mannsoni via identification of the presence and activity of elastus, an enzyme released in the parasite's larval stage, estrogen quantities in natural dietary supplements, hand and body washes, essential oils, and perfumes, and detect real-time intracellular metabolite levels. Multiple iGEM competition teams have proposed their own biosensor ideas. In 2006, Edinburgh made an arsenic biosensor whose output was a change in pH. In 2009, Cambridge's E-Chrome project engineered Escherichia coli to produce an output that was variable in color and visible to the naked eye. In 2016, the University of Sydney team made a mycobacterium biosensor that detected a food ripening hormone, ethylene, and produced chromoproteins in response. And the Ionis Paris team built an E. coli biosensor that detects toluene and benzene-based air pollutants and emits bioluminescence in response. Last year, Glasgow's Campylobacter jejuni detecting biosensor aimed at preventing food poisoning won the Food Nutrition Division. As shown, synthetic biology-made biosensors have huge novel potential applications and can provide real-time meaningful information due to their high sensitivity and specificity levels and portability, as they are smaller than and don't contain toxic or hazardous chemicals often present in current sensing analytical and diagnostic kits. Their simple operation means that trained staff aren't required, and citizens can partake in science.